So we talked about prokaryotic DNA replication initiation process. And how about eukaryotic cells? Okay, so eukaryotic cells have distinctive cell cycle, as you just said. You know, prokaryotic cells may exist a little different cell cycles, but you know, this is a story of eukaryotic cells, this interphage and mitotic phage, right? So in the prokaryotic cells, the replication takes place during the S phase, like I said, right? But then what happens during the G1 phase? Actually, that's the beginning, initiation of the replication that should happen in the G1 phase. So, 6 protein complex called origin recognition complex, we call that ORC, is made during that G1 phase, and then that is formed as an initiation replication initiator. So this complex is formed in the G1 phase, not S phase, earlier. Okay, then ORC recognizes the A element and B1 element of the chromosome, and it binds and hydrolyzes ATP. So we're talking about here ORC. Okay, recognizing A and G, those chromosomal sequence, okay, it's formed and recognized there. That's what we are talking about. A and B1 element. And ORC recruits 2 helicase loading proteins. Those are called CDC6 and CDT1, and 2 copies of MCM27 helicase. So we're talking about these guys. Yeah, so MCM27. CDC6 is described here, okay, and binds an ATP, okay. Then these 2 proteins, MCM27 and CDT1. So these are the helicase complex that's formed during the G1 phase, okay. And the ATP hydrolysis by CDT6 results in the loading of MCM27 helicase dimer. So we see that ATP hydrolysis. So then it's a dimer. It's formed MCM27 dimers and CDT2. So that's a monomer, and there's another monomer. The dimer is formed, and that is what it's saying. Okay, so ATP is required for that process. Okay. So yeah, we just talked about it, and then that's happened during the G1 phase, but at the S phase, okay, there are changes on the phosphorylation. So CDK, which stands for cyclin dependent kinase enzyme, and DDK, that's DBF4 dependent kinase. So those are both kinase enzymes. Kinase are the enzymes what doing phosphorylation. They can put phosphate onto their substrate. That's what they do. Okay, so CDK and DDK, those enzymes are activated, probably through a signal transduction pathway, and DDK phosphorylates MCM27 helicase, and CDK phosphorylates 2 other proteins. The results binding CDC45 and GNS to MCM27 helicase. So yeah, so I guess the phosphorylation is not described in this diagram, but you know, the phosphorylation is taking place by the kinase enzyme. So let me see, is it described here? Yes. So yes, there's CDK, you know, cyclin dependent kinase phosphorylating these guys. DDK also phosphorylating other proteins. Okay, so a bunch of phosphates are added to this helicase complex. Now they are described there. Okay, so the phosphorylation is taking place during the S phase. That's what he is saying here. Okay. So where are we? Yeah, right here. So CDC45 MCM27 gene complex is the active form of the helicase complex. Okay, and then the interaction between the 2 DNA helicase interrupts as 1 DNA strand is ejected. So phosphorylation, and then these 2 dimers are split. Okay, so each helicase one direction, and this direction and that direction. That's what he just said. Okay, so interaction between the DNA 2 helicase units are interrupted as 1 DNA strand is ejected. Okay, so that's what they are saying here. Okay, and then what happens? 3 DNA polymerases assemble at the origin. Okay, so 3 yeah, so polymerase, there's 1, 2, 3 DNA polymerases initiated in this replication fork. That's what he just said. Okay, there are 3 polymerase complexes: polymerase alpha and epsilon, which was already there, and the polymerase delta. Trimers are engaged in the replication fork. That's what it's saying here. Okay, 3 DNA polymerase assembles at the origin, and then CMG complex, which is made out of CDC45, MCM27, and gene S. Okay, and the 3 DNA polymerase becomes a replication fork complex. Okay, so we're talking about here, that's the replication fork complex. Okay, so that's what happens. Kind of complex. There are so many players in this process. Okay, but the important thing here is the CDK, so cyclin-dependent kinase enzyme level, is fluctuating, and that tightly regulates the loading and activation of helicase enzyme. So kinase is playing a key role in activation and inactivation of helicase enzymes. Okay, so phosphorylation is the key. So CDKs are required to activate loaded helicase to initiate the replication. So although helicase is already loaded there, for them to initiate, break open the DNA, the phosphorylation should take place. That's what it means. And then CDK activity also inhibits the helicase loading. We already said that. And CDK levels are low during the G1 phase, allowing the loading, but preventing activation. It's just sitting there. It doesn't work. Okay, but entry into cell, S phase, that is coupled with a rapid increase of CDK kinase activity, that drives activation of loaded helicase. Okay, but preventing the new helicase from loading. So the loading takes place during the G1 phase. Okay, but the activation takes place in the S phase through the CDK. Okay, cyclin-dependent kinases. Once it's loaded, activated, there's no more helicase loaded on the replication fork. That's what it means. The high level of CDK activity prevents during the S phase, and G2 and M phase inhibits the function of ORC and CDC6 and CDT. Okay, so there are highly orchestrated regulation. That's what it means. Okay. And then at the end of the replication, if you're looking at the prokaryotic genome, it's a circular right here. Okay, so replication fork is formed like ORC area in E. coli, for example, and replication will take place. It's going to go continuously, opposite direction, this direction, that direction. Let's say it's going to continue, and then you will see each template strand, the circle is replicated here, and maybe the inner circle may be replicated, located here. So now you have 2 ring structure is kind of chained together. So you have to split them. So we call that decatenation, separating 2 daughter DNA strands. So how do you separate this? You have to probably cut, and take them out, right? So that is done by an enzyme called type 2 topoisomerase. Okay, that is required to separate or decatenate daughter DNA molecules at the end of DNA replication. So topoisomerase enzyme will take care of that separation process, decatenation process. Okay, and DNA helicase unwinds the double helix using ATP, the energy from ATP. So DNA helicase can move either from 5' end to 3' end, okay, or 3' end to 5' end. Okay, you can go down, either way. So since it's going this way, on both strands going, that's what it means. Okay, so splitting, breaking up hydrogen bonds requires energy from ATP hydrolysis. And then once DNA is single-stranded, they are occupied by single-strand binding proteins. They stabilize single-strand DNA through cooperative binding. So the multimers of those proteins are binding the single-strand DNA, so they don't go back to double-strand. Okay, they bind to DNA sugar phosphate backbone without making sequence-specific contact with base. What it means is, they don't care about what bases are found. They just bind on the sugar phosphate backbone, most likely through ionic interaction. Okay, and then topoisomerase removes supercoils produced by unwinding at the replication fork. So if helicase unwinds, the ahead of the unwound side, there's more tension that's created because of unwinding. So we're talking about that area. Okay, that tension will be relieved by topoisomerase, which can nick that area and running this supercoil on the opposite side. That's how it can relieve the tension. So that's done by the topoisomerase. So not only they can decatenate, they can also relieve the tension created by the unwinding of the supercoiling. That's what it says. And then a requirement in removable RNA primers. So primers are RNA molecules, as we said, so we're talking about DNA replication process. So unless you remove primers, you have segment of RNA molecules embedded within your newly made DNA strand, right? So you don't want that. So you have to remove those primer sequences, so especially the lagging strand. Okay, there are multiple primers there. So what is the enzyme removing that RNA molecule primers? It's RNase H enzyme that's responsible for removing RNA primers. Okay, so the reason why you need to have primer is because DNA polymerase cannot start its replication without primers help. You can only grab the tail, 3' end primers, and start the replication. So primers are absolutely needed. So RNA polymerase can initiate new RNA strand on the single-strand DNA template. So RNA polymerase can make something new out of nothing. So RNA polymerase doesn't have any problem, but DNA polymerase has a problem. So that's why you need the help of RNA polymerase. That's making RNA primer. And primers, specialized RNA polymerase makes short RNA primers on the single-strand DNA template. Okay, that's what it just said. And RNA primers are used in the initiation of the DNA replication, and then they are removed and replaced with DNA molecules. Okay, so RNase H removes that primer. Okay, and then through its exonuclease activity, chewing up from the end, from 5' end. So here 5' end of the primer, it gets rid of RNA nucleotides. Okay, so once this gap is created by RNase H, then there will be DNA polymerase enzyme grabbing the tail, 3' end of this DNA template, and then synthesizing based on what it reads on the template strain. So once it's filled, and the last, the phosphodiester bond cannot be made by the DNA polymerase, because that's not a function of that DNA polymerase. That should be mediated by another enzyme called DNA ligase. That's going to form the last phosphodiester bond. So that's the entire process. Okay. So then DNA synthesis done by the DNA polymerase. Okay. So DNA synthesis requires deoxynucleotide or nucleoside if there are 3 phosphates there, and a primer. So yes, you have to have a primer tail to be able to start the DNA replication. That is what it says. So double-strand DNA template, single-strand DNA template. So DNA polymerase can read it and grab the tail, 3' end of the primer, and then you can add the new nucleotides based on what it reads off of the template strand. Okay, when it adds a new nucleotide, it uses this nucleoside that has 3 phosphates. Okay, by cutting up the phosphates. Okay. So here's the process: extension of 3' end of the primer and energy from phosphate. Okay, so here's the nucleoside. So if there's a T there on the template strand, of course it's going to grab a nucleotide, because between T and A, the double hydrogen bonds can be formed nicely within the pocket of DNA polymerase. And then the energy required for that is catalyzed by cutting 2 phosphates out. Pyrophosphate is removed, and then between phosphate and oxygen, there is that phosphodiester bond made. So that is made by the DNA polymerase. That's what it says here. Okay. And DNA polymerase uses a single active site. That's the site of the enzyme DNA polymerase to catalyze DNA synthesis. So within that active site, there's so-called kinetic probe reading that's happening. So what it means is, if the incoming nucleotide is the correct nucleotide, it can nicely make the hydrogen bonding. Okay, when it makes the hydrogen bonding nicely, and that phosphodiester bond formation is done smoothly without much resistance, right? So that's so-called kinetic probe reading. Okay, it fits in very well. But if what's coming in here is the wrong nucleotide, okay, then it cannot